The highlight of my work is this model called SimADC. It's a hybrid agent-based model that combines agent-based modeling, it's a computational model that combines agent-based modeling, ordinary differential equations, and partial differential equations, and essentially we use this model to predict the efficacy of certain ADCs. And so with the complexity of this computational model, we can look at individual cell characteristics and kinetics, drug distribution, and overall efficacy. And so we use it to study the efficacy of different ADCs and different dosing regimens. And so two of the things that we tackle or have tackled and are tackling with this model is the distribution of ADC in solid tumors and also the relationship between ADCs and the immune system. And so for the first point, we find that in vivo experiments in high expression environments, ADCs tend to localize around blood vessels, and typically something that's typical of ADCs is poor intratumoral distribution in those environments. And so in order to mediate that, our lab has pioneered this idea of co-administration with a carrier dose, which is essentially a naked antibody, so the same monoclonal antibody backbone as the ADC, but without the payload. And so when you dose them together, you create this partial competition for binding sites that pushes the ADC further into the tumor to improve intratumoral distribution, which in turn improves efficacy. And so we can see that in tumor staining images, we can see that in efficacy plots in vivo mice studies. With the model, we kind of can predict how much carrier dose do you need to get the best efficacy. And so our findings from that model is that we want to essentially dose to saturating the tumor, saturating the receptors in the tumor. And so if you reach all of the cells in the tumor, you essentially achieve the best efficacy. And the second problem that we are looking at is the relationship between ADCs and the immune system. So studies have shown that in immunocompetent mice or ADCs dosed in immunocompetent mice have more efficacy than immunodeficient mice. And so we want to know what role the immune system is playing in ADC efficacy. And so using the model, we have things like T cells and cancer cells that have this relationship in the model, and so we study that relationship. And we're looking at different things like T cell activation, so the ADCs are activating the T cells, which can kill more cancer cells. We're also looking at the influence of macrophages in macrophage ADC uptake and payload release, and things like free payload release in the blood. And so this relationship between immune cells and ADCs is very complex, and we're using the model to kind of narrow down why some of those relationships and those changes in efficacy happen.